mechanisms of amyloid pathology and predict clinical trial results. Alzheimer's disease, or AD, is the most prevailing cause of cognitive decline. Post-mortem, extensive neuritic plaques consisting mostly of insoluble fibrillar protein amyloid-beta can be observed in the brains of AD patients. Multiple familial forms of AD exist, and they are known to be correlated with mutations in APP gene related to expression of A-beta protein. But the most prevalent form is sporadic AD, which is not connected to specific mutations. Now, early appearance of A-beta pathology in the brain can be diagnosed using the measurement of A-beta concentration in cerebrospinal fluid or in plasma, as well as using positron emission tomography to reveal the accumulation of insoluble A-beta in the brain. A-beta targeted therapy is now represented as the inhibition of A-beta production using gamma secretase or beta secretase inhibitors, or activation of clearance of A-beta using immunotherapy by antibodies to amyloid-beta protein in fibrillar or prefibrillar form. Most of them has already passed through unsuccessful clinical trials or under investigation for the prevention of Alzheimer's disease. Usual experimental model is transgenic mouse carrying mutations in APP gene, but sometimes success in preclinical trials is not translated to the clinic. Many studies have formulated the hypothesis of toxicity of multiple forms of A-beta, soluble or insoluble forms, intracellular, extracellular, high molecular weight, low molecular weight. In fact, high molecular weight oligomers or protofibrils are now considered as precursors of mature fibrils and considered as being more toxic. The key mechanisms which is related to pre-on-time propagation of the disease is the secondary nucleation mechanisms, which means that pre-existing fibrils or protofibrils provide the surface for formation of new oligomers. There are two key toxic forms of A-beta, A-beta 40 and 42, which differ by 2 amino acids, and usually it is considered that A-beta 42 is much more toxic. But it has been shown in the in vitro studies that actually interaction between these two forms is also very important for toxicity. Usually fibrillar oligomers can be measured, mature fibrils can be measured using theaflavin T fluorescence. But it has been shown that if we mix A-beta 40 and 42 in equal proportion, the fluorescence will be minimal, which means that there is no mature fibrils, but some specific oligomers. And it has been shown that these oligomers are the most toxic form, as have been shown in vitro data for cell cultures measured by effect on neurid lengths, as shown by green dots, or cell viability. Previously, we have already developed a model of A-beta pathology in collaboration with Pfizer several years ago, and it has been published. Model successfully described such biomarkers as A-beta and cerebrospinal fluid, positron emission tomography markers, and biochemical data, but it has serious limitations. First of all, it didn't consider high molecular weight oligomers as an important intermediate step in aggregation. Additionally, when we described the disease progression, we used empirical time-dependent functions implying that there is some external driver for disease progression. Moreover, in the last years, more and more clinical data appear, and also clinical trials are unsuccessful. We still can draw important information about the dynamics of the system. So today I will demonstrate how we will overcome these limitations using all available in vitro informational aggregation, using information from preclinical models and transgenic mouse to reveal the mechanisms, and using information from clinical and different therapies. At first, to be able to describe polymerization of A-beta in vitro, we upgraded our existing model of polymerization because it couldn't describe heteropolymerization. For this goal, we adopted an approach from chemical kinetics, where copolymerization of different substances successfully described, and now we consider not concentration of fibrils, but the concentrations of different types of the growing ends, such as, for example, A-beta 40 or A-beta 42. We also incorporated secondary nucleation effect, which means that existing fibril provides the surface for the formation of new dimers or other protofibrils. We were able to describe in vitro data successfully, so you can see that under all different conditions with different proportions of A-beta 42 to 40, we are able to describe both a high rate of formation of fibrils as well as absence of mature fibrils with the equal proportion of A-beta 40 and 42. And secondary nucleation effect has also been described satisfactorily because it has been shown that addition of fibrillar oligomers to the solution actually decreases the rate of polymerization, because fibrillar oligomers seed their own formation but not accelerate formation of mature fibrils. We also used in vitro data for monoclonal antibody MAB158 to reveal the mechanisms and check whether our understanding of mechanism will allow to describe in vitro data. We suppose that this antibody can block the growth of protofibrils as well as it prevents the secondary nucleation effect. We were able to describe by our in vitro model the data about the decrease of formation of fibrils, which was measured also by thiaflavin T fluorescence. And our model also predicts lower level of aggregation, but we also were able to describe the absence of significant decrease of initial rate of aggregation. To describe the in vivo processes, we applied approximately the same structure that has been published, but we added the processing of APP inside the brain cells. So we also considered that some oligomers, including high molecular weight oligomers, can be formed and further released to the extracellular space, where they can maturate to the insoluble plaques. We also added glial processes to describe the uptake and degradation of insoluble A-beta and oligomers, because according to some publications, pharmacological effect of some immunotherapy is really activation of glial cells. This is a complete scheme that I will not stop by, but it shows most of the processes and reactions of the model. When the model will have been recompiled, we checked whether it can describe previously used data, such as baselines in wild-type mouse, in plasma, in brain, or in cerebrospinal fluid. But we also added, as I promised, new data for pharmacodynamics. You can see how a model describes pharmacodynamics of BACE inhibitor in the brain. To translate the model from wild-type mouse to a transgenic mouse, which overexpress mutant human APP, we simply had to increase the rate of production of A-beta in our model, changing the parameters for APP and base processing. It allowed to describe successfully the levels in cerebrospinal fluid and plasma of the mouse, but unfortunately it couldn't allow us to describe the accumulation of soluble and insoluble A-beta. At first, we used the feedback, the most important feedback from A-beta to A-beta degradation, supposing that toxic species significantly inhibit degradation of A-beta. But even complete degradation didn't allow to describe such high accumulation as 10,000 times in the brain. So it became clear that we also need to use another feedback, a significant increase of amyloid production because of A-beta toxicity. We also had to change the form to be able to describe the shape of curve when the accumulation of soluble and insoluble forms starts at about 5 or 7 months. When we have changed the form of function depending on insoluble A-beta to hazard function and introduced the activation of production of A-beta, we were able to describe accurately the accumulation of insoluble forms in the brain together with soluble forms. Regarding the human data, we also used for calibration for the healthy subjects pharmacodynamic data. Here you can see data for beta secretase inhibitor, verbicistat, but we used data only for one dosage, and the other dosages were used for validation, and we can see that model correctly describes dose-dependent increase of the effect for A-beta 42 in cerebrospinal fluid. The data for semaglutide have been used only for validation of the model, but not for calibration, and we suppose that it illustrates well how our mechanistic approach allows to describe the data, the independent data set. To describe the disease progression data, we used just the same mechanism as it had been used for transgenic mouse, and it allowed us to describe the accumulation of insoluble A-beta as well as the decrease of A-beta42 in cerebrospinal fluid, which is usually observed. But for human, we had to consider some contribution of age-dependent function, because in human, the concentration of A-beta is not as high as in mouse. The data for soluble A-beta have been used only for validation because it is most unclear measurements, and concentration of soluble pool strongly depends on the methods of obtaining the substances. And so we only were able to reproduce in the model the increase of soluble pool in the brain with the disease progression. And finally, the most interesting part is the predictions by our model of long-term treatment. For verbitostat chronic treatment of transgenic mouse, model predict quite similar results for the treatment of aged mouse, about 20% decrease of A-beta42 in the cortex. And for soluble pool, it predicts the values quite similar to what we do see in cerebrospinal fluid. It's interesting that if we start therapy in this mouse earlier, then the level of inhibition for insoluble pool will be better. But this is not the case for the soluble pool, and so final level does not depend on the age of therapy start. We also tried to describe the data for transgenic Swedish mouse, which differs a little bit from the mouse that have been used for calibration. But we were able, in principle, to describe the data for preventive study when the mouse is 8 months old. We can see the decrease in protofibrils and similar decrease with respect to placebo in our model, as shown by blue curve. Also, the decrease for insoluble total pool was underestimated by our model. Regarding the curative treatment of the aged mouse, model correctly predicted the absence of the effect for total insoluble pool, which is shown by correspondence of black and red curve here. But this can be overcome for the earlier treatment. But the model couldn't predict the decrease of protofibrils, which means that not all transgenic mouse are the same, and as our model was calibrated to predict the very high level of soluble pool, then it cannot be used to predict the levels for this mouse, which has much lower level of soluble pool than the TG2576 mouse. And finally, the data for human. We used our model to simulate the clinical trial analogous to what had been published not long ago. We have taken from the report the baseline characteristics for the patients, the age, and positron emission tomography SUVR. And we only calibrated the model by only one parameter, maximal effect for microglia activation, which can allow to clear the insoluble plaques. And we can see that indeed model describes quite well the dose-dependent increase of effect on SUVR during the 80 months, which is similar to the data. We do not know what is going on in the brains with protofibrils in AD patients, but the model predicts that they change very well also by about 50 or 60%. Regarding the simulations for the later stage disease, for example 75 years, model predicts absolutely no effect for insoluble pool and predicts significant effect for protofibrillar pool, which may allow to decrease toxicity, but unfortunately it doesn't achieve the level of healthy subjects. So in conclusion, I would like to point again that we use all available in vitro, preclinical and clinical information to inform our model, and in fact it appears that really indeed transgenic or preclinical models can be described without any additional time-dependent drivers, but using only A-beta. Still the model is semi-empirical in progression part, because although we consider dependence on A-beta, we do not describe the exact mechanisms. And we suppose that for human additional mechanisms can be included because a lot of biomarkers have been measured. Analogous model for tau pathology will be presented on our posters on Wednesday and QSP social event. And for further investigation of this system, we are now organizing Alzheimer's disease modeling consortium to consolidate the efforts of the team from different pharmaceutical companies and to reveal the new mechanisms, biomarkers, and collect them all into the comprehensive Alzheimer's disease platform. Thank you for your attention.